Hi, I'm Charlie Hotchkiss. I've been working in the Fern Lab at the National Tropical Botanical Garden here on Kauai since October of 2019. I'm very excited to share with you the research we have been working on in our lab. This research, in a nutshell, is all about figuring out how long it takes to grow ferns from spore to sporophyte and getting specific timelines for different species. So you might be asking why? Why does it matter if we know the timelines for spore to sporophyte growth for native Hawaiian ferns? I'll tell you. Ferns are often overlooked in habitat restoration projects in the Hawaiian Islands, even though they make up more than 1/4 of the native flora. Their function in the ecosystem is not fully appreciated, and they are not readily available to most organizations. The results of our experiment, although varying greatly by species, emphasize that growing ferns from spores is a lengthy process, and planning ahead for numbers is vital in restoration planning and conservation management. The small amount of data we've collected so far indicates that most fern sowings need to be done 2 years or more before planned outplantings. By repeating this experiment with the same species, we can begin to define average sporophyte growth times. Knowing timelines for growth from spore can enable planners to include more ferns in outplantings. So I'm going to give a brief explanation of fern biology just so we can all understand what I'm talking about. As many of you may know, ferns are pteridophytes, meaning that they are vascular plants that reproduce sexually through spores. Some ferns do reproduce asexually, but the majority of ferns reproduce through spore in a process we refer to as alternating generations. Ferns spread their spores. Those spores turn into small green discs called gametophytes. The gametophytes produce the sex cells. When there's moisture, the sperm can swim through the water to nearby gametophytes. If fertilization occurs, the chromosome is going to double, grow a sporophyte. That's going to mature into a fern, make spores, start the process over again. Here's a list of the species that we used in our experiment. These are all Hawaii endemic species, some of them being single island endemics. We have over 30 species growing in the lab, and we randomly chose these 7 for our experiment. Our hypothesis is that the rate at which gametophytes and sporophytes grow is different from species to species, and that we can narrow down those timelines and give specific averages for spore-to-sporophyte production for each of these species through repeated experiments. In this slide, you will see our data sets we got from the experiment. As you can see from the table, 2 of our species in our experiment have not produced any sporophytes yet. This isn't something we are overly concerned about, since we have had other culture trays within the Fern Lab lay dormant for long periods of time before producing sporophytes, and we have also successfully propagated both of these species before. Something else we are looking at in this experiment is when is the most productive window for our culture trays. You can see an example here of those data sets in this slide. Something we have observed in the lab is that all of our culture trays seem to produce sporophytes slowly at first and then ramp up. Then they have a window of time where the culture tray is highly productive and then start to slow back down. We are trying to figure out how long that window of high productivity is and when it starts for each species. So at the end of our experiment, we have some numbers and loose timelines for growth in these species, but it's a one-off, and we all know that scientific findings usually don't hold much weight until you have the same results over and over again through repeated experiments. What is pertinent to the end goal of actually defining the timelines for each species is repetition. This means we need to be consistently propagating these 7 species and collecting the data and comparing it with past data sets. We are constantly tracking all movements of plants in our lab and nursery for our inventory system. So data is being collected on a regular basis, which means we keep track of all the dates for sowing, transplanting sporophytes, ferns that die, which makes compiling data for research that much easier. Our research is not only valuable to our garden and conservation efforts here in Hawaii. This experiment outlines how others can replicate the same kind of research with any fern species. I would encourage all botanical gardens that have the resources to start a fern propagation facility to do so. Our garden was able to outplant well over 10,000 native ferns since 2010. With a deeper understanding of the growth timelines in these ferns, we could easily double that number for the next 10 years. Imagine if other botanical gardens joined the efforts. We could restore native ecosystems one fern at a time. Thank you for listening. I'm honored to get to be a part of this conference and to be representing the National Tropical Botanical Garden. I'm happy to answer all questions and inquiries. Please send them to my email at chotchkiss@ntbg.org. Enjoy the rest of the conference and have a ferntastic day.